So now we are going to see about nodule formation, how the nodule is formed. So here multiple interactions between rhizobium and roots of the host plant take place.
Okay.
Rhizobium microbes, which are symbiotic association with the legume plants. So they have multiple interaction between the roots of the host plant. Okay. So this rhizobia, which is nothing but the rhizobium. So this rhizobia, which is nothing but the rhizobium, they multiply and colonize on epidermal and root hair cells first in the soil. They multiply and colonize first the root hair cells of the root. Okay. So this root hairs, due to the colonies of this rhizobium, they curl inwards. They curl like this inwards, and these rhizobium invade the root hair. They invade the root hair. Okay. So this infection, now it is known as infection thread after invading root hair and the full of rhizobium bacteria. This stage is known as infection thread. So now bacteria is carried to the cortex cells of the root. Cortex are the next cells of epidermis. Okay, next to epidermis, cortex cells are present. So here in cortex cells, the bacteria are travelled after the infection thread. Okay. So here in cortex cells only, they initiate nodule formation. Okay, nodule formation in cortex cells. They flow from hairs to the cortex cells. Okay. So this is known as nodule formation. And this nodule formation is directly has vascular connection with the host plant. Vascular is nothing but the xylem connection. So they travel and reach the xylem and connects. Okay. So these have direct vascular connection with the host plant. So this nodule have enzyme. This nodule have many enzymes. So enzyme nitrogenase and leg hemoglobin. That's why it is pink in color at the center. And nitrogenase is composed of molybdenum and iron compound. Okay. Molybdenum and iron compound is of nitrogenase. Okay. So these both are proteins. What these do, these convert atmospheric nitrogen to ammonia. Okay, so these are present in the nodules of these plants. Okay, so generally the equation is here. This one: nitrogen plus 8 electrons plus 8 H plus hydrogen plus 16 ATP is the energy. Adenosine triphosphate gives rise to 2 molecules of ammonia plus H2 plus 16 ADP. Now adenosine triphosphate becomes adenosine diphosphate. Okay, plus 16 inorganic phosphates. Okay. So this is the equation generally. Here 8 electrons and 8 hydrogen atoms. 8 electrons and 8 protons are required. H plus is nothing but the protons. Okay, 8 electrons and 8 protons are required. Okay. So this molybdenum iron compound is made up of the nitrogenase is made up of molybdenum iron compound. Okay, that is what is known as nitrogen. So this is how the nodule is formed, and the bacteria travel inside it, and it has that vascular connection with them and converts the atmospheric nitrogen into ammonia. Okay, so this is the mechanism taken here. So next we will be seeing some more characteristics of this nodule formation. Okay, and about ammonia and transamination we will be discussing. Okay. So now we are going to see about nitrogenase and its activity.
Okay.
So these nitrogenase are composed of molybdenum and iron complexes. So this nitrogenase is sensitive to oxygen. Okay. Sensitive to oxygen means in the presence of oxygen it cannot convert nitrogen to ammonia. In the presence of oxygen.
Then it will be so less than,
No. See, first it requires anaerobic conditions. Without oxygen conditions it requires. So there are some oxygen scavengers are present inside the root nodules called leg hemoglobin. So this leg hemoglobin protects these nitrogenase from oxygen.
It's like a layer. Okay.
So that is the process of oxygen.
That's the use of oxygen.
Okay. So that is why it is known as oxygen scavenger generally. Okay. So this leg hemoglobin protects this nitrogenase from oxygen. Okay. So now we are going to see about reductive and animation. Okay.
Animation.
Reductive and amination. I am sorry. So reductive amination and transamination under it is divided into because what ammonia is converted into? Is it converted to amino acids or what? So how it is converted after entering the plants? So based on that it is divided into two: reductive amination and transamination. Okay. So in reductive amination, ammonia reacts with alpha ketoglutarate acid. Okay, it is an acid, alpha ketoglutarate acid, and it produces glutamic acid.
Glutamic acid.
Okay, glutamic acid is nothing but an amino acid. Okay, so it is converted into amino acid. And then now transamination. So transamination is nothing but transfer of amino group of one amino acid. In amino acid there are amino groups.
Yeah.
So that amino group is transferred to the keto group of keto acid.
Keto group of keto acid.
Yes. Okay. So that is a chemical compound. So this amino group is transferred to the keto group of keto acid. Okay, keto group. So now it produces. This generally occurs in only glutamic acid. Okay, this amino acid is generally glutamic acid. Okay, and now. So along with transpiration, when transpiration occurs in leaves, along with transpiration, the nodules of some plant, okay, nodules of some plant, for example, soya bean plant, or the nodules of that plant, export the fixed nitrogen as ureas. Ureas are substances which have high nitrogen to carbon ratio.
After the process it exports.
Yes.
Okay.
So this is what about reductive amination and transamination. So they have given specific equation in the book where the equation represents the amino group of one amino acid is transferred to the keto group of keto acid.
Okay.
So this is the general process, simple process taking over in reductive amination and transamination. Okay. So we have seen about ureas also, which have high nitrogen to carbon ratio. It is very important. So with this the chapter ends. Okay.
Thank you for watching.
Please subscribe, and if you have any doubts, please leave that in the comment section.
If you like this video,